Assalamu alaikum dear students. Today we will discuss Lab Instrumentation MCQs Part 1C. Sandwich ELISA is used to detect option A antibody, B antigen, C antigen plus antibody, and option D complement. So the correct option is B antigen. Here is a very short explanation. The sandwich ELISA is designed to detect antigens by using two antibodies. Sandwich ELISA is particular for antigens. For example, hormones, cytokines, viral proteins. Subscribe now and press the bell icon. Never miss an update from a TECHNOLOGIST. PCR is used, option A, site specific recombination, B, site directed mutagenesis, option C, site specific translocation, and option D, both A and B. So the correct option is B, site directed mutagenesis. Explanation: PCR, polymerase chain reaction, is most commonly used in site-directed mutagenesis to introduce specific mutations into the DNA. All of the following are thermostable polymerases except option A, TEC polymerase, B, Wnt polymerase, C, DNA polymerase 3, and option D, PFU polymerase. So the correct option is C, DNA polymerase 3. So explanation है, DNA polymerase 3 is the bacterial replicative enzyme and is not thermostable, unlike the other options. PCR is used to dash option A, amplify gene of interest, B, construct RAPD map, option C, detect the presence of transgenes in an organism, and option D, all of these. So the correct option is D, all of these. Explanation: PCR is not limited to amplification. It supports various applications like genetic testing, forensics, and research. This is so important MCQs. The first step of PCR is option A, denaturation, B, annealing, C, primer extension, and option D, none of these. So the correct option is A, denaturation. ELISA measured the concentration of antibodies or antigen in option A, CSF, B, pleural fluid, C, blood, option D, synovial fluid. So the correct option is C, blood. Explanation: ELISA is versatile, but blood remains the gold standard for the most diagnostic and research application. ELISA was introduced in option A, 1992, B, 1971, C, 1994, and option D, 2001. So the correct option is B, 1971. Here is a very short explanation. ELISA became a gold standard for detecting proteins antibodies due to its high sensitivity and specificity. It replaced older methods like radioimmunoassay (RIA) because it avoids radioactive labels. Thank you for watching my videos.